or it could flop this way, or it could flop that way. So it could flop kind of like, it's almost like it's rotating in a circle. It could flop here, here, and here. Right? And as it flops in different places, the reaction is going on. Right? So the process of catalysis involves the rotational floppiness of lipoic acid rotating around here and letting things happen in each active site according to what each active site does. Okay? Does that make sense? So, you know, here we have the diameter of the particle. It's quite large, of course. There's 60 E2s in it, just to give you a sense of the size. All right. So the long flexible arm is the key to activity. So this is then, if we look at E1, E2, E3, right, this is a schematic of how the overall pathway works. And so when we talk about channeling, what we're talking about is pyruvate coming on here, a reaction occurring here by which CO2 is split off. Thymine pyrophosphate is a 2-carbon carrier, and then it moves the 2-carbon onto, right, from here, this 2-carbon unit gets moved onto the lipoic acid as the lipoic acid is flopped onto E1. Right? Then the lipoic acid moves onto E2, and there's a transacetylase reaction, so that the 2-carbon unit is transferred from lipoic acid onto CoA. When it's transferred onto CoA, it makes this high-energy thioester. I mean, it was a thioester before, it's a thioester now. Right? And so then, this has gone from being oxidized to being reduced. Right? As it picks up the 2-carbon group, you have to break that bond, right? So it basically amounts to reducing that SS bond. Right? And then once it's reduced and it's transesterified off, so you have 2 SHs, it has to be reoxidized in order to come back. Right? So if this is reoxidized, then it gets to give its electrons to something else. Right? So the second redox reaction is here, so that these SHs become oxidized and FAD becomes reduced. FAD is not diffusible, as you know. It's bound to this E3 protein, and the E3 protein binds FAD, converts it to FADH2, binds NAD, and carries out, catalyzes an electron transfer by which these electrons go here. So the ultimate electron acceptor of this complex is NAD. So electrons are ultimately coming from pyruvate as pyruvate gets oxidized, and they're going to NAD. Right? So it's always important to keep that in mind if you're looking at this is a complicated thing, a bunch of different steps. But if it's a decarboxylation, right? It's also oxidative, as pyruvate is getting oxidized into acetyl-CoA. Okay, and you can see that here. This carbon-carbon is turning into a carbon-sulfur, so COC goes to COS. Right? So this is oxidative. Not only is CO2 split off, but pyruvate is oxidized. And because pyruvate is oxidized, something else gets to be reduced. And so what is that thing that's getting reduced? First it's lipoic acid, then it's FAD, then it's NAD. And so it's like you have this little electron transport chain, if you will, like this little transport of electrons between cofactors or among cofactors all on the surface of this enzyme. So here's the steps are written out in a little bit more detail here. Substrate channeling, because we're moving this 2-carbon unit across the surface of the enzyme. It's not as if we're forming something, letting it dissociate and come back. We're keeping a covalent intermediate. So we talk about it as substrate channeling. It's not quite the same use as substrate channeling as a different example would be. For example, in glycolysis, we have 2 separate polypeptides coming together, and the product from enzyme 1 transfers directly to enzyme 2 without dissociating and coming back. That's the conventional way of talking about substrate channeling. But your textbook talks about PDH as also involved in substrate channeling. So we can broaden the use of that term to also mean something like this. Is that clear to everybody? I talked about channeling last time in terms of how it might work in glycolysis. glycolysis, for example. Yeah.
Just to interrupt that, so it's basically the swinging arm that allows it to be considered a channeling mechanism.
Yeah, that's right. I think that's right. If we look at, I'll just put this back up. Oops. So reactions that are not channeled, an enzyme binds the substrate, if it converts it to product, it releases the product binds the second enzyme, right? That's what we usually talk about. But if you have a complex between E1 and E2, and typically channeling requires that kind of non-covalent complex between the enzymes in the pathway. So the pathway is first E1 and then E2 for catalyzing the successive steps. Then once the product is formed on E1, it moves across the surface, or it moves through a tunnel to get to E2, and then the product of E1 becomes the substrate of E2. And if you have another enzyme like E3, also attached on and non-covalently linked into a bigger particle, then the product of E2 becomes the substrate of E3. And so you can see how that might be efficient because you don't have to have dissociation and reassociation events. Question?
Is there importance in the complex being non-covalent?
Not really. I mean, it could be mostly probably for regulatory reasons. If it's non-covalent, it's easy for it to come on and off. Okay. It's not that common for proteins that are doing separate functions in cells to actually be covalently linked to each other. Usually the interactions are non-covalent. Yeah, and because they're non-covalent, it also makes them hard to study because you break open a cell and you imagine that you might study how glycolysis could be channeled, maybe for part of it, but all those interactions among the glycolytic enzymes get broken up once you've dispersed the cytoplasm into your big test tube with 0.1 molar tris. Right? At that point, you've lost the association probably, because this kind of channeling association is often very weak. So there's this kind of channeling, and here, we could say it's channeling in the sense that we have 3 different polypeptides that are not covalently linked to each other. E1, E2, and E3 are not covalently linked to each other. The basis for the channeling, though, is a covalent attachment of part of the substrate after CO2 is lost, and then that gets moved through. So the substrate doesn't have to diffuse through a tunnel or something like that to get from E1 to E2. It just gets carried over because it's covalently linked. Okay? So it's kind of like a channeling in which there's a covalent linkage as opposed to a channeling where the substrate, the product that's formed in the first enzyme has to diffuse. Okay, so here are the steps. First, pyruvate gets decarboxylated, so CO2 comes off right away. And thiamine pyrophosphate, as we saw before, can carry a 2-carbon group. Right? So this requires splitting the carbon-carbon bond. So the positively charged thiazolium ion is going to stabilize the carbanion that forms in that carbon-carbon fluid reaction. Then the lysine, the reduced form of the lipoic acid, which is called lipoyl lysine, because the lipoic acid is covalently bonded to, covalently attached to the end of a lysine. So sometimes that unit is called lipoyl lysine. That picks up the 2-carbon unit, which then gets transferred to acetyl-CoA, and then the reduced form of lipoic acid basically has to be regenerated. So steps 4 and 5, product is formed here, so you can say you're done. And steps 4 and 5 are basically regenerating the enzyme. And the process of regenerating the enzyme means reoxidizing lipoic acid. And of course that means you get to reduce ultimately NAD, and that's good because that means NADH can go off into the electron transport chain. Questions? Okay, so we have some new principles. We have channeling, we have a couple new cofactors, we have a very large enzyme with many subunits.